Hi and welcome back. I apologize. I believe our last bit of video at the very end cut off short because I kind of exceeded the time limit on my video, but you got the gist of it. We were talking about the ER to the Golgi and how the proteins mature as they go through. The very last thing we were talking about is how proteins leave the trans side of the Golgi apparatus in a transport vesicle. By this point, they've been matured, they've had all their modifications necessary, they have their molecular tags, so they know where to go, and how that metaphor for going through school is very equivalent to what's going through with a protein as they mature through the cisterna of the Golgi apparatus, the cisternal maturation model. So that's where we left off. And we're still in the endomembrane system within all these organelles of the cell. The cell is very complex. You are a billion cells that are going on in your body right now. So there's a lot of work that needs to be done, and every little organelle is doing its job beautifully. So we have 2 more organelles to go through on the endomembrane system, and then we're done with organelles, and then we'll talk about a couple other things inside the cell as well. So let's pick it up where we left off. Number 4 in our endomembrane organelles that we were talking about for the endomembrane system. Number 4, these are going to be called the vacuoles. So what the vacuoles are, these are just literally membrane-bound maintenance compartments. You always have to have some kind of maintenance going on in any structure or building that's going on. Same thing with your cells. So these are membrane-bound maintenance compartments. Now there's 3 different types of vacuoles depending on what kind of species you are. So for us, in animal cells, you have something called food vacuoles. So little a, in animals, you have food vacuoles. And you guessed it, they help store food. So what happens in a cell, so if here's my cell, and I'm just showing you a little bit of it here. So here's my cell, here's my nucleus, and I got my ER, my Golgi, and all that. So outside the cell is some food, some well-digested food. So it's like in tiny macromolecule size by this point. What's going to happen, and I'll blow up this chunk of food right here so you can see what's going on here. These are enlarged pieces of food. This food is going to get in contact with the cell membrane. The cell membrane is actually going to take in the food, and it's going to form a food vacuole. So there's my food chunks that got in there. So this would be my food vacuole. This process of taking something in to the cell is called endocytosis. So this happens in animal cells. Now, this looks a lot like a transport vesicle, but it's not. It's actually a food vacuole. If you look on the inside of this membrane, what's on the inside, the fluid that's on the inside, is whatever was out here. In a transport vesicle, when you're going from ER to Golgi and all that, these little transport vesicles are taking the fluid that was in here and moving it out and moving it here. It's inside the cell, it's intercellular fluid. This vacuole, the fluid that's in there, is extracellular fluid. It's from the outside. So it has a different function, has a different structure. It's doing something totally different. So we call these vacuoles in animal cells. So what you need to know for food vacuoles is that food enters into the cell via endocytosis and creates a food vacuole. Just something coming in is called endocytosis. The word cytosis is movement that's going all around. Endo means in. You could assume the opposite would be exocytosis, but when we're taking in food, it's endocytosis. So that's for animals. Next, in plant cells. So you got animals, and now you got plants. In plants, the vacuole is called It's actually it's a little bit different. It's called a a central vacuole. So central vacuole. So if you were looking at a typical plant cell, they usually have these nice little geometric shapes, and here is the nucleus for reference. What you would see is this huge vacuole. It takes up the majority of the space inside the cell, because what's going on in there is it's storing tons of the fluids inside the cell. Inside here, it contains saps, pigments. So if you have a red rose, that's where it's at. Red pigments, you could have toxins in there. It just depends on the type of plant it is. Also can hold water, holds water. And these can expand or contract depending on the tonicity the plant cell is in. If you go back to our last unit on water, remember if you have a hypertonic or a hypo or hypertonic or an isotonic solution, plant cells are going to behave differently. It's this central vacuole that's going to either expand or contract to modify the tonicity for the cell. So in plants, you got a central vacuole. It contains all these lovely things inside. And lastly, in protists, our lovely single cellular eukaryotic cells, they have something called a contractile vacuole. So a contractile vacuole sounds exactly like what it does, but what's happening here with the contractile vacuole, these guys are pumping water in and out to maintain their tonicity. So if you have a cute little paramecium kind of thing, and it has its little nucleus and it's going to have its contractile vacuole, depending on what the the concentration of solutes in the solution it's swimming in, water is going to go in or out of this contractile vacuole accordingly to keep this guy alive. So contractile vacuoles will expand and contract to maintain tonicity for the cell. So that's vacuoles. Got three types: food vacuoles in animals, contractile vacuoles in plants. I'm sorry, central vacuoles in plants, and contractile vacuoles in protists. All right, one more vacuole. But before we go to the next vacuole, I want to bring your attention back. I'm sorry, one more organelle. Before we go to the last organelle, I want to bring your attention back to the food vacuoles, because we're going to use this guy to set up the next organelle. The next organelle is called the lysosome. And what the lysosome does, it's a hydrolytic compartment in the cell. So I want to take you back to Unit 1 really quickly. If you're doing a hydrolysis reaction, I just want you to realize how biology is continuous and you can't forget what you learned before. It's going to come back. When you're doing a hydrolysis reaction, that's when you're breaking down the polymers into little monomers. And when you eat food, you're eating these huge chunks of food that get broken down into macromolecules. And those macromolecules have to be broken down into monomers so we can actually use them for building things inside our cells, whether that's energy for literally building proteins or fats or whatever it is. So these food vacuoles are taking in these large macromolecules, and the next organelle is actually going to help process them for us. So number 5 and last organelle. You guys have been great. The last organelle is called the lysosome. Lysome. S-O-M-E, sorry, lysosome. These are hydrolytic compartments in the cell. And the thing about the lysosome, it's a highly specialized organelle. It contains tons of lysosomal hydrolytic enzymes. So it contains hydrolytic enzymes. To break down macromolecules. So the way these function, I'm going to bring another color in. So if we go back to our cell, and so here's the food. So here's my food vacuole. All right, inside the cell, I'm blowing this up for the sake of teaching it. Inside the cell is this organelle. This is your lysosome. The lysosome is actually going to fuse with the food vacuole. So you're going to get this half lysosome. They're going to come together and fuse together. And the food that's in the lysosome, the food that's in the food vacuole will now be digested by the enzymes in the lysosome. And eventually, the lysosome is going to eat everything up in there, and it's going to release all these monomers out into the cells so they can get used to do whatever they need to get used to do. So, the way this works is the lysosome fuses with the food vacuole to digest the macromolecules. Now, as we've gone along in medicine and biology, we've discovered that there are some really sad lysosomal storage disorders that are out there, and if you're lacking enzymes that don't break down a specific macromolecule, that macromolecule can build up, and depending on the cell types they're in, they can do some really sad damage. So having good lysosomes is key for nourishment of your cells and ultimately your body, but also maintaining that proper balance to not let a buildup of macromolecules occur inside your cells as well. Okay, so that's a short lesson. I wanted to stop there so you could finish your organelles. So we've went through all the organelles inside the cell. We started with the cell membrane, which we did last unit a lot, but we got into the nucleus, the mitochondria, the chloroplast, all these wonderful organelles that do so many beautiful specific functions inside the cell. And now we ended up with the lysosome. Come back for our almost done with our cell section. We're going to talk about the different, this is called the cytoskeletal matrix of the cell. This is how, if you want to think of it in one way, it's kind of like it lays the tracks and the roads for inside the cell. So these things know how to move within the cell. The cell is like a city, and things have to get from one point to another point. So there's tracks that are laid, there's roads that are used, there's things that help maintain the shape of the cell. We're going to talk about all that. So come on back, we're going to talk about the cytoskeletal matrix, and we'll be wrapping up our cell section very soon.